Hello all, let's look at question number 5 from GATE Biotechnology 2018 paper. So the question is, which one of the following is an example of a neurotoxin? Option A, cholera toxin. Option B, streptolysin O. Option C, botulinum toxin. And option D, diphtheria toxin. Let us now look into the details of each of these toxins and the mechanisms of action. The first one is cholera toxin. It is produced by Vibrio cholerae, the bacteria which causes the disease cholera, causing severe diarrhea and dehydration, usually spread by water. So the cholera toxin is an oligomeric complex made of 6 protein subunits. These subunits include a single copy of the A subunit and 5 copies of the B subunit. So both these A and B parts of the subunits are connected by disulfide bonds. So the 5B subunits form a 5-membered ring that binds to GM1 gangliosides on the surface of the intestinal epithelium cells. So the A1 portion of the A subunit is an enzyme that ADP ribosylates the G proteins, while the A2 chain fits into the central pore of the B subunit ring. So this is the overall structure and the structure of the cholera toxin. It binds to its receptor. The complexes is now taken into the host cell through the receptor mediated endocytosis. So inside the cell, the disulfide bond is reduced and the A1 subunit is freed to bind with the human ADP ribosylation factor 6, which is ARF6. So this binding exposes its active site, allowing it to permanently ribosylate the G's alpha subunit of the heteromeric G protein. So this results in constitutive cyclic AMP production, and this further leads to the secretion of water, sodium, potassium, chloride and bicarbonate ions into the lumen of the small intestine, which leads to rapid dehydration. The next one is streptolysin O. So before that, streptococci are normal flora found in the upper respiratory tract of humans and animals. So some of them, like Streptococcus pyogenes, may be pathogens causing infections. So it produces non-suppurative lesions, which means that there is no pus secreted. So they also cause acute rheumatic fever and glomerulonephritis, which occurs following the infection. So glomerulonephritis is the inflammation in the filtering part of the kidney, which is the glomerulus. So Streptococcus pyogenes produce different toxins, which are hemolysins or streptolysins, erythrogenic toxins, streptokinases or fibrinolysins, and deoxyribonucleases and hyaluronidase. Let's now just look into hemolysins. So Streptococcus pyogenes produce 2 distinct hemolysins termed as streptolysins O, which are oxygen labile, and streptolysin S, which are serum soluble. So both of these streptolysins lyse erythrocytes, which are the RBCs, polymorphonuclear leukocytes and platelets by forming pores in the cell membrane. So streptolysin O belongs to a family of hemolysins found in many pathogenic bacteria. Here's an overview of the mechanism of the cytolytic toxins and other coenzymes produced by Streptococcus pyogenes. So the pyrogenic exotoxins produced by the bacteria cause various effects, including the rash seen in scarlet fever and streptococcal toxic shock disease. And then streptolysins, the streptolysin O and streptolysin S, damage the mammalian cells, resulting in cell lysis and release of lysosomal enzymes. And then streptokinases catalyzes the conversion of plasminogen to plasmin, causing the lysis of clots, facilitating the rapid spread of these organisms. And then enzymes like C5a peptidases, they inactivate the complement component C5a and hence blocks the immune responses to the pathogen by the host. And then streptodornases, these are DNases that degrade the viscous DNA in necrotizing tissue or exudates, aiding the spread of the infection. And then hyaluronidases disrupt the organization of ground substance, facilitating the spread of the infection. So these are the functions and the mechanisms of each of the toxins and the enzymes produced by Streptococcus pyogenes. And we also saw what the streptolysins do. And the next option was botulinum toxin. And just to have a background, let's learn about botulism. So botulism is a rare but a very serious paralytic illness caused by a neurotoxin that is produced by the bacterium Clostridium botulinum. So botulism can present in three forms, mainly characterized by the method of entry of the bacteria into the human host. So it could either be foodborne, and foodborne botulism arises from eating foods that contain botulinum toxin. And then infant botulism, most prominently presented from the ingestion of honey, which contains the Clostridium botulinum spores, which then can germinate in the intestines and release toxin. And then wound botulism is when a wound becomes infected with Clostridium botulinum. So the foodborne botulism is the most common one, and it has caused outbreaks in the past. So the Clostridium botulinum toxin is categorized within the category A bioterrorism agents, mainly due to its toxin's great lethality. And organisms within this category are considered to be deadly pathogens. And as stated by the CDC, the category A pathogens have the greatest potential for adverse public health impact with mass causalities, and most require broad based public health preparedness efforts like improved surveillance and lab diagnosis and stockpiling of specific medications. So these category A agents also have a moderate to high potential for large scale dissemination or a heightened general public awareness that could cause mass public fear and civil disruption. So botulinum toxin is one of the toxins categorized into like bioterrorism threat. So this table basically explains the symptoms and signs, and these are the same for wound botulism. So, however, the gastrointestinal issues like nausea, vomiting, abdominal pain, cramps, discomfort, diarrhea, and fever are typically absent in wound botulism. So, the symptoms of foodborne botulism typically present within 18 to 16 hours after eating a contaminated food, but can occur from anywhere between 6 to 10 hours post ingestion. So, the main way to diagnose botulism is look for symptoms of botulism in mice that are injected with a sample of the patient's serum or stool. So, testing for botulism is typically performed by the health department laboratories. So, the botulinum toxin consists of 7 different serotypes A to G based on their variability in structure, target site, and toxicity. So, all the 7 types work by the same general mechanism of inducing muscle paralysis by preventing the release of acetylcholine from the presynaptic neuron at the neuromuscular junction. So, since they differ in the targets, their individual effectiveness in inducing flaccid paralysis is variable. However, botulinum neurotoxin A is the most potent of the serotypes and therefore has been used most frequently in research and in therapeutics. And uh, the A and B serotypes remain the only serotypes that have been manufactured and approved by the FDA for commercial use. So, the toxin is a deadly substance where 1 gram has the potential to kill 1 million humans. So, it has been used for medical and cosmetic purposes called the Botox treatment, like for face feature correction. So, here are the 7 serotypes, their cellular substrates, and target protein cleavage sites. So, the botulinum neurotoxin serotypes cleave at least one of the 3 intracellular substrates that are essential for neurotransmitter release. So, SNAP25, syntaxin, and VAMP interact with each other to form a multimeric complex that mediates neurotransmitter release. So, this table clearly shows how the botulinum neurotoxin serotypes target parts of this key complex and that they even cleave specific residues if acting upon the same target site. So, when neurotransmission fails, the brain cannot send signals to the muscles and hence the muscles can't move, which is basically paralysis. So, botulinum toxin is also used in cosmetic indications. So, the Botox injections are shots that use this toxin to prevent a muscle from moving for a limited time. So, these shots are often used to smooth wrinkles on the face. They're also used to treat neck spasms, sweating, overactive bladder, lazy eye, and other conditions. So, Botox shots also may help prevent migraine. So, the medicine in Botox injections is made from the same toxin that causes botulism, but the forms of purified botulinum toxin used by licensed healthcare providers meet the medical control standards. So, these standards were FDA approved, and as a rule, these botulinum toxins used for medical purposes are not harmful if they're used correctly. So, here's an example of how the toxin could be used for cosmetic purposes, and they could be used in the upper face to treat horizontal forehead lines and to treat nasal crouch lines, and even in other parts of the face to treat nasal flare, poppy chin, and brow lift, and to treat facial asymmetry. Like most of the models, they use Botox treatment to make their face look more symmetrical. So the injection of 4 or 5 sites equidistantly along the equator of the forehead with a total of 60 units of the Botox toxin is done, and the patient is shown before treatment in the upper panel, and 1 week after the treatment in the lower panel you could see that these horizontal forehead lines are gone. So, like we just saw before, the toxin can also be used for non cosmetic purposes like to treat neck spasms, sweating, overactive bladders, lazy eye, and other conditions in several fields like dentistry, in gynecology, in gastroenterology, and so on. The next option was diphtheria toxin, and diphtheria is an acute bacterial infection caused by Corynebacterium diphtheriae, and this is a gram positive bacterium. It secretes a potent exotoxin, which is the major determinant of the pathogenicity. So, that is the diphtheria toxin. And diphtheria is endemic in India, and it's very common below people of the age 15, and it's common in winter and autumn seasons, and both males and females are equally affected. So, the pathogen spreads through droplet infection during coughing, sneezing, and talking. And once infected, the infection remains till the virulent bacilli are present in lesions, usually for up to 2 to 4 weeks. And diphtheria is a rapidly developing acute febrile illness with both local and systemic pathology. So, the primary lesion in the upper respiratory tract leads to necrosis of the epithelium, and severe injury leads to plasma leak, fibrin network formation with bacteria, and a pseudomembrane formation, and which is adherent to underlying tissue like nose, pharynx, larynx, and tonsils, and thus the infection leads to scraping and finally leads to bleeding. So at this site, they produce toxin, like at the bleeding site, it produces the toxin that is absorbed and disseminated to the whole body. So these exotoxins affects the heart, kidney, liver, spleen, muscle, peripheral nerves, and adrenals. So, to conclude, diphtheria is a serious bacterial disease that causes severe inflammation of the nose, throat, and windpipe, usually the upper respiratory tract. Now, coming back to a question, which one is an example of a neurotoxin? So, we saw that cholera toxin leads to gastrointestinal infection and dehydration, and streptolysin causes hemolytic conditions, and botulinum toxin is a neurotoxin causing paralysis that answers a question, and diphtheria toxin causes respiratory tract infections and inflammations. So, botulinum toxin is an example of a neurotoxin.